Welcome to our lecture on the reproductive system. And as usual, we have three parts to this lecture. Essentially, the outline includes an overview of the physiology of the reproductive system. And we will focus on both the male and the female reproductive system. And then five specific phenotypes or events, which are fertilization, implantation, pregnancy, paturation, and lactation. The learning objectives, at the start, I want you to be able to describe the role of the sex chromosomes and sex hormones in sexual determination and differentiation. And then to describe the role of these hormones and how they're controlled during the development of male and female reproductive systems. And specifically look at the events of fertilization, pregnancy, and paturation in the context of hormonal control. For learning outcome one, we'll be looking at the role of the different chromosomes and hormones in the sexual determination and differentiation. Just briefly describe the events in meiosis and its significance in the process of making our sperm and eggs, which is gametogenesis. And then, of course, the process itself. And finally, to describe the hormonal regulation of the reproductive system. Humans are sexually dimorphic, which means males and females are physically distinct. And this distinction is usually defined by the sex hormones that also control our behavior and influences our brain development and response to various different situations. And the human fetus that grows from a zygote, right, when the egg and sperm fuses, is actually driven by specific sex hormones that ultimately will influence many different aspects of our adult life. So when we look at the components of the reproductive system, you have the gonads. And so the gonads are essentially what contains the germ cells, which are essentially stem cells that produce the sex cells as well as the hormones. And then when we differentiate it between males and females, the male gonads are the testes, which make sperm, and the female gonads are the ovaries that make eggs. And besides that, we also have the internal genitalia, and these are all the glands and the ducts that essentially move specific fluids or cells. And then you have the external genitalia, which is essentially how the specific cells are transmitted between males and females. I'm sure many of you have seen this. It's called a karyotype. So it's not that our cells are very well arranged. In general, this is actually an artificially produced map of the chromosomes that contain all the genes in our body. And we know that each of our cells, apart from our sex cells, are basically diploid. What it means by diploid is that we actually have one for each given chromosome or each given gene. We actually have two copies, one from our father and one from our mother. And so we have 46 chromosomes, 22 pairs of autosomes. That's chromosome 1 to chromosome 22. And then you also have a pair of sex chromosomes. And as you know, males have two X's, and you can see this X chromosome is so much bigger than the Y chromosome. And if a cell contains X and a Y, then you know it is a male. So, and essentially the sex chromosomes a person inherits will determine the genetic sex of that individual. But we of course now know that we are talking about the biological or genetic basis of sex determination, we now know that gender and sexuality and sex, is not as binary as it is conventionally seen. But from a genetic standpoint, a female is defined as the absence of a Y chromosome, and the male is defined by the presence of a Y chromosome. And that's because we have males who have two X's and one Y, and this particular disorder called Klinefelter. And then you also have women who have one X only, and this is a particular syndrome called Turner syndrome. And both individuals are viable, but they suffer from reproductive as well as certain other abnormalities. But in general, okay, for the most part, a female offspring will be two X's, and a male offspring will have an X and a Y. And you will notice the difference is that these cells, the eggs and the sperms, they only have either an X in the case of eggs, but in the case of sperm, they can either be X and a Y. And that is why the determination of sex is actually cannot be blamed on the woman, but actually on the man. Well, you can't blame him too. He doesn't pick and choose which sperm swims faster, but essentially the determination of the sex of the baby is actually going to be by the type of sperm that is fertilizing the egg. We all start from a single cell and then sort of divide into the trillion cells that we are. And the reason why a single cell can become so many different types of cells is because the single cell, the zygote, is totipotent. And it's the most potentially differentiable stem cell, which can then become any type of cell in the body. And as the cell then, starts dividing and this a fetus is formed, essentially the cells will move from a fully totipotent to a pluripotent to a multipotent. In the context of multipotent, it means that it can differentiate to some types of tissues, but not all types of tissues. And finally, to a unipotent, where, like for example, the stem cells of the intestine lining will generate as much interstitial lining, but it cannot become, suddenly the lining of the alveoli or what have you. So when we talk about the sexual organs, during the embryogenic stage, so basically as you are developing, and especially in the first 6 weeks of existence after fertilization, actually your reproductive tissues are bipotential, which means they can develop into either a male or a female reproductive structures. So that's right, boys. Actually, all of us at some point were females until a specific gene was switched on. And this gene is the SRY gene. And this gene is located on the Y chromosome. And the SRY gene is called the sex determining region on Y chromosome. That's why SRY. And so that is why, basically that when individuals that have a Y chromosome, but if the SRY gene is not switched on during the fetal development, what will happen is that you will be having a Y chromosome, but you might not have male genitals. And you may have heard of a few athletes who have lived all their lives thinking they are women until they get genetically tested. And then they find out that they are at least male genetically. And so you have a few athletes who have had this issue, and it's a big ethical concern. And you probably have heard of Caster Semenya and a number of athletes in the same. So these are not transvestites or transsexuals, but these are individuals that actually, do not have any male, sort of genitalia, but they have the male YX chromosome. So this critical gene, the SRY gene, and it's switching on usually around the 6th week of embryogenesis, is extremely critical. Because it's this gene, when it's switched on, that will then facilitate the development of the gonads, which is your testis. If you don't have it, then it will continue to grow into ovaries. And then the various different ducts. The Wolffian duct, which is derived from the embryonic kidney, that later on develops into the vas deferens, and that's essentially how your semen is actually transferred into your penis. And then the Mullerian duct, which is essentially derived from the embryonic uterus that then develops into the fallopian tube. So this is an image of what I just described, how at 6 weeks, if those cells contain the Y chromosome, the SRY gene is switched on and makes SRY protein products. And the presence of these protein products will result in, number one, the embryo directs the medulla to develop into a testis. Yeah, the bipotential gonad develops into a testis. And then the specific anti-Mullerian hormone from the testis will degenerate the Mullerian ducts. So therefore, these don't become fallopian tubes. And finally, the testosterone from the testis will convert the Wolffian ducts into a seminal vesicle, vas deferens, and the epididymis of the the epididymis, essential for the transmission of the semen and the sperms and ejaculant material. Whereas in the case of a female, because the SRY gene is not switched on, the gonadal cortex becomes the ovary, and then the Wolffian duct degenerates, and the Mullerian duct becomes a fallopian tube, the uterus and the upper part of the vagina. So this is essentially the pathway in which the SRY gene directs the male development. So exactly as what I said, it stimulates essentially the production of testosterone and also the anti-Mullerian hormone. And there are two types of cells, the Leydig cells and the Sertoli cells. And these Leydig cells, they secrete testosterone. The Sertoli cells secrete the anti-Mullerian hormone. And the external genitalia also, differentiates. So as you can see, where the clitoris is, becomes a penis, and then where the labio, sort of swelling, labia minor and major, essentially becomes the scrotum. And so these are all analogous. And that's why we talk about how these tissues, depending on the presence of SRY and later on testosterone as well as the anti-Mullerian hormone, will also drive the changes inside and outside. Isn't it fascinating how it's one particular stimuli that ultimately triggers the differences that we actually see, that is obviously larger than we could ever imagine. So what is the gene that, when expressed at 6 to 8 weeks during embryogenesis, that directs male reproductive development? That's right, SRY, the sex determining region on chromosome Y. What is the hormone that is expressed that will continue to direct male reproductive development? Very good. Testosterone is the hormone, right? So the next part of this lecture, we'll be then talking about the cells, that is actually produced by the specific gonads. And the process is gametogenesis. And essentially, it actually happens during our development into a fetus and into a baby. And then at some point it stops, and then it resumes again during puberty and continues to adulthood. And that's because of the expression of these specific hormones. And this process is timed differently in males and females. As you know, our age of our puberty or onset of our menstrual or development of secondary sexual characteristics are later in men. But this varies and also is very dependent on the diet. And as you know, both of these hormones, estrogen and testosterone, are all secreted, in the form of cholesterol. So they are actually essentially cholesterol derivatives. And so childhood obesity and the various different diets that we actually eat has been one of the reasons why the age of onset of puberty is lowering in each generation. So let's go back again to looking at on a cellular level. And at a cellular level, we talk about the importance of cell division, duplication as a basis of how sperms and eggs are actually made. And the cells of the gonads undergo mitosis. So mitosis is your typical cell division where the DNA of your cells actually duplicate, and then the cells divide, and then the DNA separates. So basically the new daughter cell's DNA have the same DNA content as the parental cell. But in the context of meiosis, it's a little different because now the daughter cells actually have half of the DNA of the parental cells. And this is really important because you imagine, right, if your sperm and your egg cells actually was exactly the same in the context of DNA content, it also had 2 copies of everything, then the baby that is going to be born will have 4 copies of the DNA. And you can imagine that the amount of DNA that we're going to accumulate in every generation is going to be a lot. And so to ensure that we only have 2 copies for each and 1 copy each from our parents, this cell division, meiosis, actually happens, where the gamete is no longer diploid, it's haploid. And it carries half of all the genes, and it is sufficiently matured into viable cells when it is fertilized. And we will see. What you want to get out of here is just understanding the process of meiosis. And so what happens during meiosis is that, number one, the cells double in size, right? So 2 times, because basically DNA replication occurs and then you have multiple copies. I wouldn't bother too much about the process of crossing over. That's just one way of making sure that the sperm cells actually contain a combination of your grandfather and grandmother's genes. And so basically these chromosomes actually have some crossing over. But I wouldn't be too concerned. For your particular understanding now, you just have to imagine that after there has been a doubling of DNA content in the cells, what happens is, looking at number 7, is meiosis 1. And in meiosis 1, it is similar to mitosis, where now the cells divide. And when the cells divide, now you actually have essentially the same as these cells, right? So these are very similar. But another thing happens, and that is meiosis 2. And during meiosis 2, the cells further divide. So you can see these cells are half to what these cells were. Right? You can see you got 2 only, whereas here you got 4. And so this is how meiosis is different than mitosis, because you basically have 2 divisions for a given cell. And so when you look at the spermatogenesis, you can see that these are the stem cells of the sperms, and they're called spermatogonia. And as the cells divide during meiosis 1 and then meiosis 2, you basically then have maturity, you have spermatozoas. And it's exactly the same with oogenesis. Now, instead of spermatogonia, you actually have oogonia. And this oogonia then, sort of divides and becomes primary oocytes. And then what is very interesting is that when a baby is born, basically a female baby is born, they are born with set amounts of primary oocytes. And this is usually about 400 primary oocytes. And so that is the very difference between male and females. In the case of males, they continue to produce sperms, and that's why they call it as menopause. But in the females, basically, they are born with a set number of primary oocytes. But what is interesting here is basically the primary oocytes don't undergo meiosis until they actually reach puberty. And meiosis 1 only occurs during ovulation. And have a look here. During meiosis 1, what will happen is that only one egg will actually be chosen to continue to grow. The other degenerates into what is called a first polar body. And this secondary oocyte will only undergo meiosis 2 if there has been fertilization. And during fertilization, basically another polar body is created, and you actually then have an ovum that later on becomes a zygote once it's actually been fused. So this is the distinction between oogenesis and spermatogenesis. The completion of meiosis 1 and meiosis 2 is dependent on the ovulation and fertilization process. As I've already alluded to you, the process in which the sperms and the ovums are actually produced by the testes and the ovaries are driven by hormones. And these hormones actually come from the brain as well as the endocrine cells in the gonad itself. And the sex steroids, i.e. the testosterone and estrogens, because they're all cholesterol derivatives, they're very closely related to one another. And when you hear the word androgens, generally, you're talking about the androgens in males, which is the testosterone that comes from the testes. And when you talk about, the dominant hormone in females, then you will hear estrogen, and especially estradiol too. But you probably would have heard of progestins and also androgens. So what is important is basically there is some testosterone still being expressed by females. The only difference is the amount that they express is a lot lower, and it is these differences that differentiate the characteristics of a male. On top of that, as you know, the adrenal gland also secretes some small amounts of sex steroids, and these are your androstenedione or your DHEA, which are also hormones that influence the amount of testosterone and estrogen that's actually available in the body. So this slide talks about how all of these, essentially have an origin and a precursor in cholesterol. And obviously there's some blank boxes here, and they're just intermediate compounds. And you can see how, this cholesterol, is essentially used to make progesterone, testosterone, DHT, estradiol or estrogen, cortisol, corticosteroids, and aldosterones. The control of both males and females hormonal production in the testes and the ovaries actually is on the basis of a hypothalamic pituitary gonadal axis, the HPG axis. So which means something that's secreted by the hypothalamus controls the pituitary glands, and the pituitary gland secretion controls the secretion of the gonads. So, but luckily the nomenclature for many of these hormones are very, very consistent. So for example, what is the hormone that actually is released by the hypothalamus? They are the gonadotropin releasing hormones. because they are tropin. So tropin means growth. So the growth of the gonads, gonadotropin releasing hormones that are released by the hypothalamus. And essentially, this will control the secretion of the anterior pituitary gonadotropins, which are the luteinizing hormones, LH, and the follicle stimulating hormone, FSH. So these two anterior pituitary hormones will then act on the gonads to initiate and maintain the gametogenesis, especially we will be looking at it in the context of the ovarian and uterine cycle in females. And what this LH does is that it acts on the endocrine cells to secrete steroid hormones that will actually facilitate the initiation and maintenance of gametogenesis by FSH. This particular signaling pathway really captures how there is this whole hypothalamic, pituitary, and gonad axis. So at the hypothalamus, when there is an internal environmental stimuli, the gonadotropin releasing hormone is expressed that then stimulates the secretion of LH and FSH from the anterior pituitary gland. And this LH and FSH has an impact on the gonads. So FSH is follicle stimulating hormone that then tells the testes and the ovaries to start producing gametes. LH in females only will also support the gamete production, and LH will also stimulate the endocrine cells to increase the steroid and peptide hormones. And the presence of specific steroid and peptide hormones, so when there's an increase in testosterone and estrogens, they will then loop back. The short loop will go to anterior pituitary and say stop expressing, but it'll also have a long loop feedback back into the hypothalamus to tell the gonadotropin releasing hormone to essentially stop if the amount is enough. If the amount is not enough, then this presence of this testosterone will stimulate it to have more, and so there will be a positive feedback. There's also a short loop negative feedback, and this is directly from the pituitary to the hypothalamus. So you have the H, P, G. The feedback can be from P to H, or also from G to P and G to H. And so the feedback loops, as I had mentioned, are really critical in maintaining homeostasis. And besides the gonadal hormones and the pituitary hormones that can produce the short and long loop responses, the FSH secretion also introduced the expression of inhibins and activins. And these inhibins and activins are hormones that or proteins that ultimately also inhibit the secretion of FSH. So it goes back to the anterior pituitary and say, hey, stop or increase its secretion. So what is the cell division process involved in the production of ovum and spermatozoa? That's right, it's gametogenesis that's driven by meiosis, right? But because if you notice, the cells do divide and actually have double copies before at meiosis 1, so there's also an element of mitosis that happens earlier. But meiosis is the main process. What are the hormones that you've learned thus far, and where are they made, and what are their functions? That's right. Remember the HPG axis, which is the gonadotropin releasing hormones from the hypothalamus that stimulates the anterior pituitary to then release FSH and LH. That one stimulates gametogenesis, another one secretion of endocrine hormones, and then you have estrogen and testosterone. Sorry about the spelling error here for estrogen. Right? You've got to put an N, estrogen.